Welcome to the latest installment of the AST Journal Club series. Today's Journal Club features the JCI Insight article, Tregs Integrate Native and CAR-mediated costimulatory signals for control of allograft rejection. This session is sponsored by the AST's Community of Transplant Scientists. In a moment, I will turn the session over to our moderator, Dr. Scott Krummey from Johns Hopkins University School of Medicine in Baltimore, Maryland. Before we begin the main presentation, we have a few housekeeping notes to help you engage with today's session. Currently on your screen, there is a viewership polling question displayed for the audience. Please take a moment to answer the question while we finish the remaining announcements. If you have any questions for our panelists during the Journal Club, we encourage you to participate by using the Q&A button in the Zoom webinar panel. Please note that any questions submitted via chat window may be missed during the presentation. This Journal Club is being recorded and the archive will be available on the MyAST website in 2 to 3 business days. And finally, when you log off at the conclusion of today's Journal Club, you'll see a link for a short evaluation survey to complete. We ask for you to complete this survey to help us keep our content current and engaging. I will now turn the session over to our moderator, Dr. Scott Krummey, to begin our presentation.
Thanks, Rafaela. So I'd like to welcome everybody to this exciting AST Journal Club. So we're very lucky to have with us today Dr. Megan Levings, the senior PI in this study, as well as the first author, Dr. Isaac Rosado-Sánchez, who will be presenting his work. So by way of a brief introduction of both of our speakers, so Dr. Levings is a professor in the Department of Surgery in the School of Biomedical Engineering at the University of British Columbia in Vancouver. She's the Canada Research Chair in Engineered Mean Tolerance and the lead of the Childhood Diseases theme at BC Children's Hospital Research Institute. She's internationally recognized for her work in human immunology and leads a group of trainees and staff who are researching primarily how to use T regulatory cells to replace conventional immunosuppression in the context of transplantation and autoimmunity. She's the recipient of numerous awards, including the 2020 YWCA Woman in Distinction Science, Research and Technology, the 2022 Simon Fraser University Outstanding Alumni Award, and the 2022 UBC Faculty of Medicine Distinguished Researcher in the Basic Science category. So research in her group is focused on the role of T regulatory cells, including understanding the molecular and cellular biology of Tregs, elucidating their role in transplant tolerance, cancer, and inflammatory bowel disease. Dr. Rosado-Sánchez obtained his PhD in molecular biology, biomedicine, and clinical research from the University of Sevilla in 2018, where he studied Tregs in HIV-infected patients. He continued to develop his interest in Tregs and how they could be used in cellular therapy and transplantation in Dr. Levings' lab starting in 2018, whereas we'll hear about today, he's been studying how to optimize CAR-Tregs to increase their safety and efficacy as a cell therapy. So as I've mentioned already regarding the topic of the work today, cellular therapy and CAR T cells in particular are really an exploding area overall in medicine. And as far as the use of cellular therapy and transplantation, Dr. Levings' group has been a leader in the development of CAR-Tregs for this application to try to induce transplant tolerance. So on behalf of the AST Community of Transplant Scientists, I'd really like to welcome again Dr. Levings and Dr. Rosado-Sánchez and thank them both for taking the time to present their interesting work today. And as Rafaela said, please pay attention and enter any questions you have into the Q&A, and we'll hopefully be able to get to them after the session, after the talk. And so with that, I will turn it over to our speakers, and thank you again for your time.
Thank you very much, Scott, and to the AST COTS members for selecting this paper for today's Journal Club. It's really an honor to be selected. I'm just going to do a quick little intro and then hand over the main session to Isaac. So as Scott mentioned, my lab has been working on how to use regulatory T cells as a replacement or less harmful adjunct therapy to immunosuppression in the context of transplantation. And after our initial paper in 2016 describing the potential benefit of using CAR-Tregs, we realized that we needed to take a deep dive into understanding more about the functional biology of the structure-function relationship between a CAR and a regulatory T cell. So the first paper that Isaac is going to mention briefly that we published investigating that was all done with human cells. And as Scott mentioned, my lab mostly used to work on human cells, but with the results of that paper, we realized that we had a lot of questions that we really needed to answer in a fully immunocompetent mouse model, which brings us to this paper. So Isaac was involved in both the studies. And so he's going to tell you background, rationale, and all the figures for the paper. So looking forward to the talk. Go for it, Isaac.
Thank you so much for the nice introduction and also for this opportunity. I'm going to start, I'm going to start sharing the conflict of interest disclosure. So Dr. Megan Levings holds paid patents and license related to the A2 CAR technology, and Dr. Krummey doesn't have anything to disclose. So has Dr. Levings actually bring up, we are working in trying to optimize CAR-Tregs therapies. And if we just focus in what is the optimal CAR design for Tregs, several papers just came out in the last few years, denoting like an increasing interest in the topic. All of them reach similar conclusions, but we're going to focus in this one, functional effects of chimeric antigen receptors, co-receptor signaling domains in human regulatory T cells. So our lab has worked extensively trying to characterize different costimulatory domain CAR-Tregs in humans. And for doing that, we generate like a lot of different CAR variants carrying different costimulatory domains from the CD28 and the TNFR superfamily. We make lentivirus, transfect human Tregs, and after all the QC, we test those cells in vitro and in vivo. And when we focus in vitro, we realize that when CAR-Tregs get similar only through the CAR, a CD28 domain CAR was superior than the others in terms of inducing activation of Tregs. As we can see in this plot in dark blue, CD28 CAR induce a stronger activation of Tregs compared to the others, but also inducing proliferation, stability of Tregs, and suppressive function. On the other hand, when we tested in vivo, we use a graft versus host disease. As you can see also in dark blue, a CD28 CAR Tregs perform much better than all the other CAR variants studied in delaying the progression of the disease. So we got a clear winner here, and we actually define a CD28 CAR as the optimal to be used in Tregs. As I said, the other studies basically reached the same conclusions, but also, as Dr. Levings brought up, we think those models has pros and cons. They are very useful, but on the other hand, has some limitation. And one of the main limitations that immune deficient mouse model have is that we are missing pieces of information. The engraftment of AP6, for example, is quite poor in this model, so we were missing how CAR-Tregs can interact with other cells to get activated, but also to induce their suppressive function. Another piece of information we're missing is the role of DSAs, of donor-specific antibodies, that we know are very important in some types of transplantation. So keeping all these limitations in mind, we wanted to step forward, and aim to study the optimal CAR design in Tregs, but in an immune-competent model of transplantation. For doing that, we generate 8 different CARs mouse variants in this case, carrying different costimulatory domains from the CD28, the TNFR family. We also generate an irrelevant control called HER2, and we generate retroviral particles and harvest FOXP3 Tregs from a FOXP3 GFP mouse strain. So this mouse has a reporter for FOXP3 GFP that allow us to harvest highly pure Tregs with the need of fixing PERV then. So we can sort FOXP3 positive CD4 live cells, transfected, and force them to express whatever CAR we are interested in. So this is how the different CARs actually looks like. We can track the CAR expression by expression of the tag CEMIC, and also with the fluorescent marker MQ2, which give us the information about the transaction. And obviously, all the cells that express the CARs were able, with the exception of the irrelevant CAR, of course, were able to interact and bind to HLA2 tetramers. So important to say is that most of the CAR were expressed, with the exception of CTLA4 and TNFR. For some reason, these CARs weren't properly expressed in mouse Tregs, even if the amino acid sequence of CTLA4 in mouse is identical from humans. And we have a lot of experience with this CAR in human, and we never got this problem with with human Tregs, but with mouse Tregs, we weren't able to make that work. So we stopped using these two variants and focusing the others. Regarding those variants that were properly expressed, we got a similar expression of those. So there were not huge difference in their level of expression. And we can see in here where I'm showing frequencies of c-MYC positive NK2 co-expressing cells. There were no difference in FOXP3 expression, nor GFP, the reporter, truly FOXP3 expression, or even co-expression of FOXP3 in Helios. All of them looks very nice. And finally, we actually rule out if these new CAR variants are able to induce HLA2 antigen-specific activation of these Tregs. And for doing that, we make a culture system in which we could culture these cells with HLA2 kappa6 and other controls. And if we focus in the red histograms, we can see that only the cells expressing the CAR, so MK2 positive, NK2, MYC positive, NK2 positive, are able to respond when cultured with HLA2 kappa6. So this CAR induces an antigen-specific response in mouse Tregs. After all the QC, everything was working fine. We say, okay, it's time to move ahead, and we start testing these new CAR variants in vitro. And what we found is that when we stimulate the different CAR Tregs only through the CAR, a CD28 containing CAR is definitely superior. Superior in terms of inducing proliferation, cytokines production in general, but importantly IL-10 and also suppressions. As we can see in this graph, where the CD28 CAR Tregs in blue performs better than all the other studied CAR Tregs variants. Sorry, I forgot to say that these results actually match to what we previously observed in human CAR Tregs and others lab also saw. So after all in vitro work was done, we moved to our skin transplant model or immune competent mouse model. Briefly, we performed HLA2 skin transplant into regular black 6. It's important to say that those are allografts, they are not xenografts. So they came from a mouse strain that expressed HLA2 and treat these mice with the different kind of CAR Tregs we generate. And here we go, the first surprise, the first interesting results of this experiment, different from what we actually observed in immune deficient mouse model and other labs also described in immune deficient mouse model. In the skin transplant immune competent model, CD28, ICOS, PD-1, and GTR CAR Tregs perform very similar. They extend the survival of the skin graft very similarly. On the other hand, a 41BB and an OX40 CAR Tregs didn't perform anywhere at all. They didn't extend the survival. Regarding the control of these different CAR Tregs of the DSA generation and levels, we focus on anti-HLA2 IgG titers, and different from the skin survival, only a CD28 containing CAR Tregs was able to reduce the level of the anti-HLA2 Ig titers, and the others had some impact, but weren't statistically different than the not treated control, the PBS mice, which is in black. It is important also to say that we track all these different CAR Tregs along the time in peripheral blood, and we actually observe from what we observe in immune deficient mouse models. So CAR-Tregs and the CAR expression on Treg in some way decreases along the time. For some reason, we don't know. So we were thinking that maybe the transduction reporter, the MQ2, which is an exogenous protein, it could be in some way driving this disappearance of clearance of CAR-Tregs or CAR-positive Tregs. So we perform another in vivo study in which we generate MQ2 positive and MQ2 negative second-generation CAR-Tregs, injected in parallel in mice, and see if the engraftment actually evolves differently along the time. Surprisingly, we didn't observe any difference in their numbers, nor in peripheral blood, nor in tissue. So we basically conclude that MQ2 is not a driver of this decay of CAR-Tregs in vivo. So we were very interested in the results, in vivo results we got. So we really wanted to understand what was happening. We wanted to rule out what is the truly contribution of a costimulatory domain in the Tregs field. So for doing that, we generate a first-generation CAR. First-generation CAR shared exactly the same structure that a second-generation CAR, but lacks of any costimulatory molecules. Being saying that, both first-generation and second-generation CAR were perfectly expressed, so we start testing them in vitro. What we got is that what has been previously actually described, that when CAR-Tregs are activated only through their CAR, a second-generation CAR-Tregs perform better than a first-generation CAR-Tregs in terms of proliferation, cytokine production, and also suppressive function. Then we move in vivo, and we got the next surprise of the study. So surprisingly, even of all the difference we got in vitro, in vivo, we didn't get any difference between the survival of the skin induced by first generation versus CD28 second generation CAR-Tregs, as we can see in red and the blue line. And if we look at the DSA levels, importantly, the first generation actually maintained the levels of DSA quite low. So they were quite decently at the same level, similar level than the CD28 second gene in controlling these DSAs. Similarly to previous in vivos, we track all the cells in peripheral blood, but this time also in tissue. And we didn't find any difference in engraftment, persistent CAR expression, activation, nor even the expression of any functional marker. So they were quite similar, actually, for not saying the same. So at this point, we were very actually intrigued for all these experiments, all the results we were getting. So we say, okay, stop. We have to go back. We have to actually try to find what others has been published regarding first generation CAR-Tregs versus the second generation CAR-Tregs, because we have in our minds all the information that came from CAR-T conventional, the cancer field. So we did that, and surprisingly, only one article was explored previously first generation CAR-Tregs in transplantation, which actually was the article that I bring up earlier. In this paper, remember, we use an immune deficient graft versus host disease, a xenogenic graft versus host disease model. We didn't actually observe any benefit of using first generation CAR-Tregs compared to irrelevant CAR-Tregs, not even the second generation CAR-Tregs. And thinking why could be truly different between this model and the ongoing models we are using in this study, we realized that this is an easy answer, right? The model we are using now is immune competent. The previous one was immune deficient. So we have a set of other cells rounding in the body of the mice that could interact with Tregs and could not only respond to them, but also give them stimulation and impact in their activation and function. So we really aim to test this hypothesis. And for doing that, we generate a new in vitro culture system where we could culture first generation, second generation, and irrelevant CAR-Tregs together with Ki-Fas-X2 all expressing HLA2 or expressing HLA2 plus CD86. Remember that CD86 is one of the ligand of CD28, which is actually highly expressed in Tregs. And the idea is to find out how Tregs get activated when we give them only stimulation through the CAR, or when we combine the stimulation through the CAR plus a native stimulation through CD28. So after doing that, we got this kind of results. This is like a summarizing figure when I show in proliferation, but we got similar results in terms of activation markers, etc., functional markers. Sorry, on WhatsApp. And the point of all of this is that if we look at the red plot, first generation CAR-Tregs increase the level of proliferation and activation when CD86 ligand is present, is available. They do two very similar ways that the second generation CAR-Tregs. In order to rule out if this is truly happening through the CD28 and CD86 axis, we add to the first generation CAR-Tregs some CTLA-4 Ig. So CTLA-4 Ig is going to block the interaction of CD80 with CD28, and when we do that, we observe that first generation CAR-Tregs decreases the level of activation and proliferation to the same levels that they got when they were co-cultured in absence of CD86. A little bit more farther, when we add CTLA-4 plus an antagonist CD28 antibody, we observe that the first generation CAR-Tregs increase their levels of proliferation to the same levels that they were, they were proliferating in presence of CD86. So the idea is that we confirm that the axis of CD86 and CD28 is working here, and most importantly, that the native co-stimulation can in some way synergize with the CAR-mediated activation to finally perform a different activation status of first generation CAR-Tregs, similar to what second generation CAR-Tregs actually reach only through the CAR. Those was in terms of proliferation and activation. We were wondering, does this actually have an impact on their function? So for answering this question, we performed a disease suppression assay. So we could culture dendritic cells together with first generation and second generation CAR-Tregs and study not only their activation, but also their suppressive function over dendritic cells. So as all we know, Tregs are able to reduce, to peel off expression of CD80, CD86, and MSC class 2 on dendritic cells. And if we look at the histogram to the left, which are the HLA2 positive dendritic cells, both kind of first generation and second generation CAR-Tregs were able to reduce the level of CD80 in comparison with the other controls and also with the HLA2 negative dendritic cells. So anyways, using this system, we realized that in vitro, first generation and second generation CAR-Tregs show also the same ability, the same function of ability to suppress CD80, CD86, and MSC molecules on dendritic cells. Again, to demonstrate that this is because CAR-Tregs, first generation CAR-Tregs, are receiving an extra help from the dendritic cells. We add CTLA-4 Ig, and we observe that when we do that, the function of first generation CAR-Tregs actually was hampered and similar to the polyclonal controls. Then we realized that all this was made in vitro. We really wanted to see if this is actually happening in vivo. So for doing that, we performed another skin transplant experiment where we did the surgery, inject the first generation and second generation CAR-Tregs, and this time we terminate the Krummey at day 7 and look at the levels of these costimulatory molecules on dendritic cells from different tissues. And if we focus on the draining lymph nodes, we observe that first generation and second generation in blue and red, respectively, doesn't have any difference. They have the same ability to reduce also in vivo the levels of these molecules in comparison with the levels of those molecules in dendritic cells from PBS-treated mice. Importantly, we only observed these effects on draining lymph nodes. We didn't observe these effects on spleen or other location, meaning that the function of CAR-Tregs is anatomically restricted to the nearby area of the graft, which is important because that reduces the immune suppression of CAR-Tregs to that area and not, to other by a standard suppression to other areas. Finally, we wanted to know if this is also happening with the other CAR variants we previously studied. So we wanted to know if the other CARs can also synergize with native co-stimulation, and if that has like an impact in their function, and we can explain a little bit better the skin graft function, in vivo function of this CAR variant. So first of all, we use the in vitro system we previously talked about, and we observed that without CD86 in the system, so the Tregs are only able to get activated through the CAR. We have like a variety in the levels of activation and proliferation of CAR-Tregs, 100% mediated by the CAR ability to activate them. However, when we occulted stem cells with CD86 GIFASX2, we found something different. So all the different CAR variants actually increase a lot the levels of activation and proliferation to very similar levels that the CD28 CAR-Tregs, meaning that in presence of CAR-independent CD28 stimulation, all type of CAR-Tregs can get activated by this mechanism similarly. But this is only activation. We wanted to see also if it has like a translation into their function of these CAR-Tregs. So we perform our DC suppression assay. I'm showing here the reduction of MSC class 2 on dendritic cells after co-culture with different kind of CAR-Tregs. And as you can see, with the exception of 41BB and OX40 CAR-Tregs, which actually were the CAR-Tregs fraction that didn't work at all in vivo, all the other kind of CAR-Tregs, including the first generation CAR-Tregs, were able to reduce the levels of MSC class 2 and other costimulatory molecules to a very similar level, so to very comparable to the CD28 second generation CAR-Tregs. And of course, this is kind of a mirror from what we got in our in vivo results, and give us a little bit of rationale of what is going on and why we observe what we observe. So to start finishing the presentation, I would like to bring the main conclusions of the study. So the message to take away is that when stimulated only through the CAR at CD28 CAR seems to be optimal, seems to be the best for inducing Treg function and activation. However, when we are working in an immune-competent model of skin transplant, a costimulatory domain seems to be dispensable. And this is because CAR-Tregs can receive other kind of stimulation from other cells that are around in that model and weren't in the previously used immune-deficient mouse models. And finally, but no less important, that is that native and CAR-mediated costimulatory signals, so CAR-mediated and CAR-independent signal can synergize to not only induce different levels of activation, but also to modulate the function of CAR-Tregs. And I think this is very important because we are always missing that part of the coin. We're always focusing on the best design of the CAR, but we are missing how other signals can impact and synergize with that CAR-mediated stimulation to finally induce like a modulate and reshape the function of Tregs. This study has also limitation. I just bring in the two principles. First of all, we got, as I said, a short persistence of CAR-Tregs in vivo, and that could be an important limitation. We don't know exactly why is this happening. It could be lower levels of IL-2. It could be the lack of coordinated antigen. We can discuss this later, but this is something to keep in mind. Another limitation is that we didn't only play around with different costimulatory domains. We also were switching transplant main domain. And we did that because we have the knowledge about what kind of transplant main domain works and get a lot better with whatever costimulatory domain. So inducing their activation, the expression and their function. So we try to keep that in order to enhance the function of the different CAR variants. So all the results we are seeing must be interpreted in terms of a different costimulatory, but also a transplant main domain. Finally, I would like to thank my whole lab, obviously Dr. Levings, my supervisor, the people that are working with us. All of them are great people, very supportive, and especially the people that are more closer to this project. Also thanks to our collaborator, Dr. Giorgio Raimondi from Hopkins University, all our funding agencies, and obviously thank you for being here, and we'll be happy to take some questions.
Great, thank you. Yeah, thank you both for that really, really interesting, detailed work. I think it's, obviously the strength of it is being able to look at, not just how things work in vitro, but being able to find the important differences in vivo systems. So I encourage everybody, again, to answer any or put any questions they have into the Q&A session, and I will be happy to relay them. So in the absence of any of those right now, I can go first. I had a question. I know, I'm very well versed in our group of the skin transplant model, and it's has a lot of advantages. But I was wondering about the potential, the difference you showed in the DSA and the reduction in DSA with the CAR-Tregs, and in the absence of having an easy model of B cell and antibody dependent rejection. I was wondering if you guys have any thoughts on whether the CAR-Tregs are functioning differently in the suppression of antibody formation versus graft rejection in a kind of T-cell dependent skin graft model. So for example, do you have any thoughts of whether they're directly interacting with the B cells, or whether the mechanism of CAR-Treg suppression is through the APCs that's also secondarily impacting the B cells?
Yeah, unfortunately we cannot give us like a certain, a certainty on this, but we definitely have information. In our previous AGT 2020, we actually explore how second generation CAR-Tregs can impact on the onset and levels of DSA. So we truly know that happens. The differences we observe in terms of control of DSAs between the different costimulatory domains, CAR variants, and finally the rejection is going to the line of what you're saying, that the mechanism involved in controlling the DSAs and finally the rejection are different. And that could, that has all the rationale, right? Because on one hand, we have the humoral immune responses, and another we have the T cells. I didn't share that data in here, but you can find it in the article. We also try to correlate the levels of DSA with the rejection of the graft. Interestingly, when we treat the mice with CARs that carry costimulatory domains from the CD28 superfamily, we got like a tight and beautiful correlation between the DSA levels and the rejection. However, when we move to the CARs carrying TNFR costimulatory domains, that correlation is very poor or absent. We don't know, again, we don't know exactly why this is happening. It's something to think about, but this is what it is. It could be, if we start politicizing, I think that maybe the rejection in that group of mice is so fast that the DSA actually contribute poorly, because when they are actually increasing, the graft is already compromised. It's possible also that the different costimulatory domains induce like different expression of chemokine, right? Chemokine receptors, so they can migrate differently to different location, and for some reason, maybe the TNFRs do not allow them to migrate to the specific area in germinal center to control that. It could be a lot of reasons, but we don't have an answer, something to look about in the future.
Great. Thank you. Okay, so a couple questions coming in here. So forgive me while I read. So one of the reasons why the CD28 construct CAR T cells cause less GVHD was because the CD8 construct induces CAR T cell exhaustion more than the 4-1BB construct CAR T cells. So is that a possible reason why you saw CAR Treg persistence was subpar? Do you think that contributes to the subpar persistence in vivo?
We didn't observe any differences in activation levels in all the time points. We were able to detect the cells in peripheral blood. Unfortunately, for those specific experiments, we didn't look at the tissue, which actually would be quite informative. Maybe could give us a little more information about that, but we don't know, basically. It's a possibility.
Another question, are you planning similar experiments in a solid organ transplant model?
Yes, we are actually working on that. We are working in trying to replicate this experiment and other kind of mouse models of transplantation to see if what we saw in skin can be, translated to other kind of, because I mean, if it's true that CAR Tregs are interactive with dendritic cells, we don't know exactly where the kind of dendritic cells that actually are in the grafts could be different. So we are in the process of doing that.
I have a similar or follow-up question to that. So you're targeting in the system A2, and you have a kind of an antigen-specific graft model. So in the human body or in any full organism with endogenous class 1, it's expressed on all cells, as you know. So do you think there's any benefit to potentially, or have you thought about potentially targeting class 2 to maybe get those T cells or the CAR cells to the APC or more into the lymph node?
It's a very interesting question that actually know personally, but other person in my lab is working in. And yes, there is a lot of benefits and also potential stuff that we can, do triggering, CAR Tregs towards MSC class 2 molecules. Maybe, yeah.
Okay.
Yeah, I mean, we're working on it in collaboration with Anita Chong. We've made a CAR from her lab against BALB/C class 2, and experiments are in progress. So excited to find out what those data will reveal.
Okay, a couple more. Does pretreatment of dendritic cells with CAR Tregs inhibit their capacity to stimulate conventional allogeneic T cells? So could you pretreat APCs before infusion?
You're asking if we infuse APCs before infusing CAR Tregs? Is that a question?
So sorry, I guess the question is more, I'm sorry, I'm bungling it. So could you essentially help make tolerant or more tolerogenic DCs, I think, by incubating with the CAR Tregs, like in vitro before?
Yeah, I think so. I think it's a very affordable approach. We could try that, definitely. And we know that CAR Tregs can reshape the function of dendritic cells. We are looking at that. But we didn't try yet to infuse those APCs after interacting with CAR Tregs in vivo. But it's a good idea. We could definitely look at that.
But they definitely do become tolerogenic. So if you use those CAR Treg shaped DCs to try and stimulate other T cells, they will stimulate less of a response than just DC.
And then another question, so about immunosuppression going along with CAR Treg. So based on these data in which which CAR Treg generation would be optimal to use with CTLA-4 Ig-based immunosuppression?
This is something that we are actually working on again. I think we can anticipate that, yes, there will be like a second generation CAR Tregs, the ones that works better with combination with these kind of drugs. But hopefully we have that answer, and we can answer that question quite soon. So.
Stay tuned for the next paper.
Yeah.
And I believe Anita Chong is asking, do first and second generation CAR-Tregs have different susceptibility to co-stimulation blockade or other immunosuppressive agents with respect to survival and function? So yeah, similar question.
Yeah, let's go to the same point that CTLA-4 yes, we definitely see some differences. We are waiting to have solid data to share. So hopefully in a few months, we have a nice paper also that covered that kind of that piece of information.
Great. Okay, I think I've covered all of the questions and all of my questions. So, sorry, I'm just scrolling quickly to make sure. Any other final call for questions from the audience? So it's been a really great discussion. Thank you guys again for your work and providing the context. I think this is really exciting and a really promising area for inducing tolerance for transplant patients. I guess I'll ask one maybe more open-ended question regarding translation. So what do you see as the next steps for taking this knowledge garnered from the mouse model and competent hosts into patients?
I have the feeling that we are opening like a new feeling here because, as I said, we have been historically behind the CAR-T conventional in the cancer field, path, and we have been assuming all the facts that those articles actually have been showing, no realizing that the way that the CAR-T conventional therapy works in cancer is completely different than how CAR-Tregs work in transplantation. Different roles involve different pathways, different target cells. So I think personally that we should focus more on, we are working on, stop to assuming that what we observe in CAR-T conventional was directly translational to CAR-Tregs. And then another point is that we have to start looking at the other side of the coin. We have to understand how not only how the different CAR can induce activation or different activation on Tregs, but also how is the synergy between those signals with the natural signals Tregs are receiving, right? And how everything can finally end up in having a better or full function. I think we should start with that. We should start looking at different kind of graphs, mouse models, or transplant mouse model to see this is something that could be translational to others. As I said, trying that for, in the future, hopefully move to the, preclinical model or clinical model.
Yeah, just to follow up on that. So there are already these two trials going on with the second generation A2 CAR, which was, like Isaac said, kind of based on the idea that everything we learned in cancer, you can just dump onto Tregs and it will hold true. So, now if I were designing that trial 5-6 years later, I probably would have said, oh, we should start with a first generation CAR. So in my mind, that's probably the next place that we'll pursue in terms of translation. So first generation CAR-Tregs.
Okay, great. Okay, well, I don't see any other questions. I thank everybody for their participation. And again, on behalf of the COTS and the AST, thank you both for taking the time to share your work with us and give us insight into your very exciting cutting edge work. And I think I will turn it over back to Rafaela for our closing comments.
Thank you, Scott. AST would like to thank Doctors Rosado-Sánchez, Levings, and Krummey, and the leaders of COTS, and all of our attendees for today's session. Please remember to complete the evaluation survey in Zoom and visit www.myast.org/journal club to view our video archives and register for any upcoming journal clubs. To learn more about AST COTS, please visit www.myast.org/COPs or connect directly to the COTS Hub online. Thank you all again for today's session.